So yeah, today I'm going to talk about the other kind of bacteria, and why I say that is because in the very first Science Slam, there was this colleague of mine, Paul, who mentioned the bad kind of bacteria. So it's true, the bacteria get tons of bad publicity, like for the bad things they do, they get you sick and stuff. Let's just say, syphilis. So, yeah, this nasty guy over there causes syphilis, huge problem, but, you know, don't go into the Wikipedia page about syphilis, it's gross, just, I warned you, right? So apologies to the Casanovas out there, but I'm not going to cover syphilis and the like, right? So instead I'm going to cover the other kind of bacteria, right? The bacteria that we can actually use, like harness their abilities for the benefit of mankind and so forth and so on. It turns out, basically, there's really tons of other bacteria out there that do so much diverse stuff. So this is what we call a tree of life. So you take all the sequences out there, and you basically calculate how different they are, and you put them nicely in this plot that, you know, people in the Borg group can do, like there. Turns out that most of it is actually bacteria out there. So like the blue thing out there is bacteria. Can you hear me nicely? Because I think there is a weird thing coming from. Anyway. So the blue thing is bacteria. They're more diverse than anything else out there and can do so much different stuff, right? Like for, as an example, you have a slide changer that's not working, nothing to do with bacteria. Ah, yeah, there we go. You have this nice bug over here called Colobacter crescentis. And so this nice thing likes to, so it's like a croissant, that's why they call it crescentis, right? So this thing likes to stick to surfaces. So imagine like on top here's the surface. And how it attaches, it uses this substance called adhesin. And it turns out that this adhesin is the most powerful adhesive that we know on Earth right now. It turns out there were some geeks did an experiment, tried to pull the bacteria from a surface, actually measuring how good it can hold this adhesive, right? And they measured it like, you know, geek mode on, I believe it was 68 newtons per square millimeter, which in non-geek terms, it means that if I cover my small finger here with this substance, I can lift the weight of a car. Not that I want to do that or anything, which is, you know, we can think of applications for this, right? Another thing that bacteria do, but they're least known for, you know, you've all been to elementary school and they all teach us, you know, the importance of trees and everything and producing the oxygen that we breathe, and we shouldn't burn down the trees or cut them down to build condos and so forth, blah, blah, blah. But what they don't teach us is basically that bacteria called cyanobacteria that live on the oceans are almost equally as important. Yay. Around 30% of the oxygen that we breathe is produced actually in the oceans by this cyanobacteria. So when we talk about global warming, one thing we don't usually think about is the rise also in the water temperature that really, really affects the population of the cyanobacteria. So then, you know, you can imagine if these die or whatever, then it would be really hard to breathe, like, say, an O'Reilly smoker room, yay. So basically, continuing with the geek stuff, very recently, these guys basically put the cyanobacteria in a paper in a very ordered way and got artificial leaves. I'm sure they look nothing like that. You don't want to know how they look like. But anyway, they do the same thing, yeah? They consume oxygen and they photosynthesize in the way that leaves do. And they can give you also sugar, but they consume it themselves, so it's fine. Going forward, if you turn on psychic mode in your PubMed search, you can also retrieve papers from the future, like this will be published in January 2015. It's really useful to see if they cite you for a paper you haven't submitted yet, so it's really good. I can teach you how to do it later on if you want. And this paper goes on and on about, you know, all the useful things that we can do with bacteria that will help us with a possible mission to Mars and whatever. One of them I said already, that produces oxygen. Surely we breathe that. It's really useful. Another thing is, you know, you can get methanobacteria. You can feed them whatever rocks or whatever you find on Mars, and they will make methane for you, which you can use as fuel. If you've seen Mad Max, it's the same thing without the pigs. Yeah, and all that in a very, very weight-efficient way, which is really cool, so that you can ship things back and forth. Right, so one of the things also that is least known about bacteria, and the jury's still out on this, is that if you take all the living things out there and put them on a scale, or didn't have a scale, so I employed a CISO. So if you put them on one end and you put all the bacteria on the other end, it might be that bacteria weigh almost the same as every other living thing out there, but there's not so really hard evidence in that, sadly. But I can understand what made people think about that is because bacteria multiply really fast. So for instance, you have these two bacteria starting here, and they all take over the screen in like, I don't know, a minute or two, which is really creepy, come to think of it, because bacteria have been there for millions of years, and if they do that, then surely we would be swimming in them. Not that we don't, but anyway, they would be all over the place, right? We wouldn't have any space to breathe and live. But what happens is basically bacteria don't really multiply that all the time. They do that only in optimal conditions. So bacteria basically suffer a lot, and one of the reasons they suffer is us, which use a lot of antibiotics, which we didn't develop in that sense. Nature did, but then we're using them more and more often. So you have, I'm going to show you what happens when you take antibiotics, right? So you have then here 3 bacteria. I'm sure you all know this name, Escherichia coli. We use it in the lab all the time. This one is a very nasty bug. A colleague of mine, Manuel, uses it. This one, I guess, gives you pneumonia. I don't know. So now, eventually, we're going to zoom into the cell wall of this bacterium. So one thing to keep in mind is that bacteria are under really high pressure. Think of them as a balloon with a wire mesh outside. So this is the wire mesh, right? This is the peptidoglycan cell wall. These are gram-negative bacteria, so you have 3 layers in the cell wall. So like inner peptidoglycan cell wall and the outer membrane. And this mesh is made by proteins called penicillin-binding proteins. I guess you can imagine why they're called like that. So they make this mesh really rigid, right? That gives all the bacteria cells their shape. Every bacterium has them. This is why they're good. They're a good drug target for beta-lactams like penicillin. So beta-lactam turns out that it looks really very much like this mesh. So it fools penicillin-binding proteins into binding it, and it poisons this enzyme. They cannot make the cell wall anymore. So if you have this balloon-like thing with a mesh that has cuts, then basically it forms sort of, blebs, and then it bursts, exactly like this. So this is a microscope image of what happens when you treat them with penicillin. This is E. coli. My favorite guy is right there, I believe. So if you look, there's bacteria popping like left and right. There's a massacre happening. But this guy here, you can see the blebs right there, yeah? And then next thing you know, this guy bursts. Because they're, So if you listen really closely, you can hear them pop. No, you can't. But next time you take penicillin, this is what happens in your body. Where were we? Yeah, here. So bacteria, they figure out ways around it, of course, because otherwise they would be all dead by now, but they're not. Yes, so here we were. So they have different methods to cope with antibiotics, like penicillin. One way is that they stop taking it in, so you have there, on the left, the improved version of this pore complex. This one keeps taking it in, this other one doesn't, so you have less of the antibiotic inside the cell. You have advanced efflux pumps that pump the drug out of your cell, so it doesn't affect you anymore. And you have molecules, other enzymes that inactivate basically the antibiotics, and then it doesn't harm your PPPs anymore. They keep making this mesh really nicely, and your bacteria keeps living on happily and multiplying to take over the screen. Very fast, as they do. So what do we do in the lab? So about it, right? So you have this drug target here, but you have these other sort of pathways that have evolved around it. So you hit your target, but not very efficiently, hence the grayed out X. And then in the lab, colleagues like Hannah Rita, who is sitting right there, are doing pairwise combination of drugs, trying to get which combinations can hit one of these pathways at the same time as this, and then killing your bacteria more efficiently. This is all from my side. Thank you very much for your attention. Have a nice evening.
Thank you, George. Your name wasn't really hard after all.
Sorry?
Your name wasn't really hard after all.
It wasn't. I've gotten used to it by now.
It's okay, I'm sorry. Any questions for George?
Either you got everything or nothing. So
It's okay.
I'm equally as happy. Anyway, I'll be around for a couple beers more if you guys want to ask anything.
Maybe drink more. George, everybody.